Hello, everyone, and welcome to today's webinar, Quality Control for the Pharmaceutical Industry, Stage 3, Continued Verification in the Lifecycle of Analytical Procedures. I'm Susie Valdez of Laboratories, and I'll be your moderator for today's event. Today's educational web seminar is presented by Laboratories and brought to you by Sartorius. To learn more, visit sartorius.com. So let's get started. I'd like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you can share this webinar on your personal social media. Just click on the Social Sharing tab to let your friends and colleagues know about today's live event. If you have trouble seeing or hearing this presentation, simply click on the Support tab found at the top right of your presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers Continuing Education Credits tab. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credit. I now present today's speaker, Jane Weitzel. Jane Weitzel is an independent consultant and has been working in analytical chemistry for over 40 years for the pharmaceutical and mining companies. She is currently a consultant specializing in laboratory management systems, GMP testing, and ISO IEC 17025. She is an auditor and an educator, and Jane has applied quality systems and statistical techniques, including the evaluation and use of measurement uncertainty in a wide variety of technical scientific businesses. She has obtained the American Society of Quality certification for both quality engineer and quality manager. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Jane, welcome. You may now begin your presentation.
Well, good day, and welcome to this webinar on analytical quality control for the pharmaceutical industry. This webinar is going to focus on Stage 3, Continued Verification in the Lifecycle of Analytical Procedures. This is the stage that really the quality control laboratories use. I just want to first state that this presentation is my perspective and not that of any other organization. So the topics that we're going to cover today are dealing with the analytical quality control. Now, it's for pharmaceutical drugs, especially as it relates to lab instruments, the weighing, pipetting, HPLC sample preparation, and we'll go all through all of these in our example. We'll briefly describe the life cycle approach to analytical procedures. There's background for this. Stage 1, development. Stage 2, qualification, where the analytical control strategy is developed. And finally, we'll go through the routine use of the procedure in the QC laboratory using the knowledge from stages 1 and 2, using that analytical control strategy. Quality control laboratories are critical. Their role is to demonstrate the pharmaceutical products are consistently manufactured, safe, potent, and pure. Now, at the same time, the number of tests run by QC laboratories is increasing. Labs have no resources to waste. The life cycle approach to analytical procedures and the analytical control strategy from that allow QC laboratories to be efficient and effective. And I just want to say I've worked for this a number of years, and I am impressed with how this life cycle approach really does emphasize efficiency and effectivity. That's really an excellent business strategy as well. Now, the USP has provided a number of resources, and there is a list that is available as a downloadable file for you. That is a number of stimuli articles and supporting articles and official general chapters. So I encourage you to go and read these if you haven't already after this webinar. Today, we are going to focus on analytical control strategy as described in this USP stimuli article that you can find in USPPF 42.5. This is really relevant to QC labs because you use the analytical control strategy. Well, with the life cycle approach, the analytical control strategy provided by that approach is a key tool to ensure the analytical procedures are working as expected. The laboratory is not wasting time monitoring variables that are not important, and sources of variability are adequately controlled. What is the life cycle approach? The life cycle management process provides a framework for defining the criteria for and development of an analytical procedure that meets those acceptance criteria. The procedure then becomes part of a continuous verification cycle to demonstrate that it meets these predefined criteria over the life of the procedure. Implementation throughout the procedure's life cycle of a change management process that is based on knowledge gained during the procedure's lifetime ensures that the procedure remains fit for its intended use. And that fit for its intended use is important. We'll see that more and more and more. And how do you define that intended use is all explained in the life cycle approach. Now, key to the general approach is an understanding of overall variability, including variability arising from the analytical procedure. So this figure shows the life cycles and its stages. Stage 1, procedure design and development. Stage 2, procedure performance qualification, and that used to be called method validation. And stage 3 is that continued procedure performance verification, which comes in the QC laboratory, which routinely runs the method. Notice that box on its side on the right, risk assessment. Now, risk assessment and risk is a really key topic nowadays. You see it in all kinds of guides. And knowledge management, no point doing something and learning if you don't manage that knowledge. And the analytical control strategy is a tool for managing that knowledge. And so these are the stages, and we'll both emphasize stage 3 in this talk. Now, so fit for purpose, fit for use, fit for intended use, all these phrases. For pharmaceuticals, that means the intended use is linked to the clinical requirement. And here we see 3 arrows representing the measurement uncertainty. Of what? Well, look. When they do a clinical study, they find out the concentration that they need to use of the drug. Well, they manufacture the drug, and you've got to make sure that that is within the concentration used in clinical, so manufacturing has to manufacture to a smaller range. Then that's tested in the lab, and the lab has to test to an even smaller range to make sure that the drug is fit for its intended use. So this little picture just shows how fit for intended use works. Now, with the life cycle approach is the analytical target profile, the ATP. It's a predefined objective that states the performance requirements for the analytical procedure. And the output of the procedure is a reportable result that must be fit for its purpose. So in that previous slide, that was the laboratory and that small arrow for measurement uncertainty. The method has to meet that requirement to be fit for purpose. And this applies throughout the life of the analytical procedure. Well, the analytical target profile, it includes the target measurement uncertainty. And that is back to that picture with the clinical and the manufacturing laboratory. That's the size of that arrow. How big can the uncertainty be and the analytical results still be fit for use? That's the target measurement uncertainty. Along with the ATP tells you the probability and it gives the acceptable probability of being wrong. So, you know, risk analysis, as you worked with it, is all really around probability. How do you define a risk? How do you quantify it? It's through probability. And the ATP provides you the acceptable probability of being wrong. So as long as your method is smaller than that, you're okay. It's fit for you. Now, these three little pictures on this slide, that represents the normal distribution of reportable values. And it's not an HPLC chromatogram. It's not a peak. It's a normal distribution curve. And it shows this range of possible values from an analytical result. So that bottom little chart with the green check mark beside it, that's a good one. You've got the limits, you've got the result as well within those limits, and the normal distribution is within the limits. You've got a negligible probability of being wrong. It's less than 0.00%. While you look at the top little normal distribution curve on the right side with that red X beside it, well, there's a bias there. And it has a large probability of being wrong. It turns out to be over 6%, which is not usually acceptable. So you don't want that. And the bottom on the right, well, there's a huge uncertainty associated with that value. The range within which the true value could probably be is too big. And so that's not acceptable. And so that's how the analytical target profile relates to target measurement uncertainty. So ideally in the QC lab, you want that green check mark, the distribution curve, and you want to verify it continues to perform like that. How is the ATP worded? I find this is useful. So for an assay, and we're going to give an example of an assay. So the procedure, that's the analytical procedure, your test method, must be able to quantify the analytes in presence of whatever that matrix is over a range of A to B%, and you define that, of the nominal concentration with an accuracy and uncertainty so that the reportable result falls within plus or minus C% of that true value with at least P% probability. Now, the variables in orange are specific for each reportable result, and it's customized for each procedure and each use of that procedure. And that C is the target measurement uncertainty. Now, there's a law here. We only have a limited time. I can't go into how you calculate the target measurement uncertainty, but all those USP papers have that. But what we're going to emphasize in this webinar here today is how useful all this stuff is. So stage one, procedure design and development. A procedures needed. Well, somebody defines the fit for purpose. Usually it's the pharmaceutical technology group, the R&D group. They define the ATP, the target measurement certainty, the probability is determined. And so they design and develop the procedure. They choose a technique such as HPLC. And along with that, as they're doing their experiments on this method, they develop the analytical control strategy. So if they find a potential source of variability is a significant contributor to the measurement uncertainty, then a control is needed for that source. In stage two, design and development procedure performance qualification, well then, in this performance qualification, which actually used to be called method validation, the analytical control strategy is confirmed. And control strategy could be all kinds of steps, system suitability, trending, control charting, providing a detailed training to the analyst, including detailed instructions in your test method and your operating procedures, strict control of things like time or temperature, specifying grades of materials, or other control mechanisms. These are all part of the analytical control strategy that is confirmed during that performance qualification. And most importantly, the measurement uncertainty is evaluated in that qualification. Knowledge management. That's important, especially in stage two, to capture the knowledge from stage one and from the qualification. And that document should be documented somewhere, preferably in a qualification report. Important thing to that is to document what is not known. So they've learned a lot of things about the method, but maybe they don't know some things, and it's important to document that. This is really useful for QC laboratories because they often have to deal with that unknown. For example, in the development and validation, they could only use one lot of reagent because it was expensive or maybe only one lot was available. Well, that should be documented. Only one lot was used. We don't know what will happen when you change lots. And you mitigate the risk of using only one lot. So the QC lab knows that when you have to use a new lot, they're going to have to do something. They're going to have to monitor more closely, qualify new lot before using, or on an actual example, or something like that. Knowledge management is very important in the life cycle approach. Knowledge management and risk, they're both very important aspects of the life cycle approach. I find often that labs have done some of this, but in absence of a good system and understanding, it's isolated and disjointed. And you'll notice that the guides, the FDA guidances and the requirements and the ICH guides are on talking about risk management. Well, how does this work for that analytical procedure in the life cycle? This chart is taken from that analytical control strategy paper that I highlighted at the beginning. So you use your prior knowledge, your expertise, you develop and you do a risk assessment. You identify potential sources of variability of risk and you evaluate them. And then you decide how to control them. Are you going to reduce them? Or are you just going to accept them? What are you going to do? And that becomes your control strategy. Of course, you do a regular risk review and document that in your knowledge management. It's the QC laboratory that's responsible for the use and maintenance of the risk management during routine use of the method. And how do you do this? Well, for an analytical procedure, measurement uncertainty and its evaluation is how you manage the risk for that reportable value. There's a lot of documents on how to evaluate the measurement uncertainty, and this webinar is not about that. But briefly, we'll go through the process. So there's a number of steps for evaluating certainty. One, you identify the measurand, which includes the analyte. And then you list the steps in your analytical procedure. And from that, you can identify potential sources of the random variability in each step. Those are the uncertainty components. And you design a process that permits an estimate of each, and then you combine them all together. And that 5 to combine them all together, we'll see that equation in our example. So uncertainty is treated as a standard deviation. This is important to know. A little bit about statistics, and we all have to learn this now. And standard deviations are combined as their variances, they're squared. So the important thing is that if an uncertainty component provides less than a third of the largest uncertainty component, it is not significant. And there's the equation, standard deviations combined, or U is the uncertainty combined. So the square root of the sum of the squares. So as a reminder, potential uncertainty sources are listed here. And I think as a QC lab person, you're aware of these. And these are examples of potential sources of uncertainty. So you use uncertainty to evaluate the risk. You know, what are consequences or severity of a risk? You evaluate that. You determine it directly, the combined contribution to uncertainty using your method performance data. Now, these are the analytical procedure development qualification experimental values from repeatability, robustness, intermediate precision, all those. That's key data that you use. And from that stage 1, 2, the QC lab is the recipient of this knowledge and the risks and how to manage them. So just to summarize a little bit, the ATP defines what the requirements of that reportable value, what you're testing its use. From that, you're going to tell you the critical quality control attributes. For an assay method, it would be accuracy and precision. And that informs your target measurement uncertainty, the highest uncertainty you could have, and that reportable value still be fit for use. And so stage 3, continued performance verification of your analytical procedures that the QC lab does. So in the continued verification, QC lab receives the analytical control strategy and they use it. It's important to have a mechanism for the routine monitoring of the performance of the procedure and controlling changes to the analytical procedure. And so that's how it happens in stage 3. Now to go through this, we're going to give an example. I always find examples are the best way to learn and to illustrate things. So we have the description of caveats for our example. We're going to use a drug substance tested for potency. 1 gram of drug substance is weighed, and the concentration of the drug substance is 1,000 milligrams per gram. If you're used to percent, that's 100%. I'm using milligrams per gram because that's the proper SI units that should be used. Now, caveats for simplicity, I'm not including some uncertainty components, just for simplicity, uncertainty of the bias or long term. In a full, proper evaluation, those should be included. So here's our method that we're using for example. And it has the 5 steps: weighing, extraction, pipetting, solvent preparation, and the HPLC measurement. And notice here, there's 2 injections, so there's 2 chromatograms. And the reportable value is the average of those 2 injections. That's our method. Remember how you combine the uncertainty components? So we have 5 steps. If we determine the uncertainty from each step, we can combine them, square root of the sum of the squares. So during the design development stages, if they're well designed, you can get the uncertainty for each of those steps. And ANOVA is a very powerful term and a tool for that. We're not going to go into that now. However, it can be done. You know, I often go into labs and I look at their qualifications and they have the data. They just don't do the ANOVA. It's not that much work. So I encourage you to learn about that. So from that proper ANOVA analysis done during stage 1 and 2, it turns out that the uncertainties for each of these steps is determined. So stage 1, weighing was 2 milligrams per gram. Stage 2, sample extraction was 10. Stage 3, pipetting was 2. In stage 4, the HPLC solvent preparation, it was determined that it didn't have any impact on the uncertainty, but you did have to control the grade. So here is an example of where the analytical control strategy is to control the grade of that reagent, but there's no contribution to the uncertainty. And step 5, the measurement. Now that's the average of 2 injections was 3 milligrams per gram. So you use the formula and notice that its certainty is 1, 2, 3, but none from step 4, and then step 5. So that is the formula. And if you calculate it, squaring them, summing the squares, and taking the square root, the combined uncertainty is 11 milligrams per gram. So just notice that. How is this useful? Well, let's take a simple little example and say, oh, QC lab uses its magic wand. Every lab has a magic wand. And what if you could remove the impact of all that variability from weighing? So there, step 1 is 0. If you do that and you go through, you'll see that it has no impact on the uncertainty. So improving your weighing step would have no improvement on your method. So that's the benefit of what you can learn and the knowledge from the uncertainty steps. Also, if you look at your balance and your calibration certificate. And Sartorius very kindly provided this one so we can look at it. And you look under uncertainty in use. On the certificate, it gives you the uncertainty from the balance itself. So the first line is 1.2 grams. We're weighing 1 gram. So its uncertainty is 0.000022 grams or 0.022 milligrams. Now this is an expanded uncertainty. We'll see that next slide, using a K of 2. So the actual standard uncertainty is 0.01. Now this is much less than the 2 milligrams uncertainty for the example. So your actual balance and calibration is not a major uncertainty. You can use this to know your balance is capable. And say if it goes out of cal or something and becomes 0.02 milligrams uncertainty, that has no impact. And you can use that in your assessment of that balance. So it's very useful. Here we have, look at another change with this method. Going back, we've got our weighing uncertainty in there. What about pipetting? It's 2 milligrams per gram. What is the impact of that? Again, looking at the calibration certificate, we can find out our pipette, how critical is it to our pipetting? So you look at your calibration certificate, and there and further on in the pages, it says the uncertainty budget was calculated from the uncertainty associated with the measuring process. And it says, the reported expanded uncertainty budget is a result from the standard uncertainty by multiplication of the coverage factor K equals 2. So to get the standard uncertainty, we know we have to divide by 2. So at 500 microliters on the certificate, the expanded uncertainty is 0.63. So the standard uncertainty is 0.32 microliters. Now, our uncertainty is in milligrams per gram. So to compare the volume impact to the weight, I change it percent. So it's 0.06% relative. And the 2 milligrams per gram uncertainty for pipetting is 0.2% relative. So if you compare that 0.06 to 0.2, it is about a third. It is significant. So the pipette that you're using is important. So that pipette is a significant portion of the pipetting uncertainty, and you have to control it accordingly. So maybe you want to verify it immediately before use, or maybe you want to calibrate it more frequently. Maybe you also want to train the analysts on pipetting techniques, and there's a lot of courses on that, and the pipetting providers give these courses. So that will help you standardize how pipetting's done, and you can train the analyst initially and perhaps retrain on specified intervals. So this is an example of where the training on the pipetting would be an analytical control strategy to control that pipette, which is a significant part of the pipette uncertainty. I have seen labs do this. When I directed laboratories, I implemented some of these trainings, and you could measure the improvement in the fewer OOSs post-training. The control charts got tighter. It really was very effective, and I've seen it when I audit labs as well. And so that's why it is a significant component of the uncertainty, and it needs to be controlled. And finally, let's talk about replication. The control strategy paper has a whole appendix on replication. It's an important aspect. Well, remember that in our method, we're taking 2 readings. There's 2 chromatograms there in step 5, and the uncertainty of that, the average of the 2, is 3 milligrams per gram. Well, what happens if we only do 1 injection? We get 1 chromatogram. What would the uncertainty be? Well, we're very lucky that there is a formula, the reality in our universe of standard error of the mean, where that standard error of the mean is equal to the standard deviation of an individual result divided by square root of n, the number of replicates. So for our example, the standard error of the mean is 3, and there's 2 replicates. So the standard deviation for doing one replicate, one injection, is 3 times the square root of 2, which is 4.2 milligrams per gram. And I've given you a reference for that in the NIST handbook. Well, here we have it. Here's our method. We're only doing one injection, so now our uncertainty from the measurement is 4.2 milligrams per gram. And here we go. Remember what the uncertainty was with 2 injections? It was 11 milligrams per gram. It's the same for 1 injection. There was no benefit for doing 2 injections. There's a lot of benefits for doing 1. You could change your analytical procedure to use only 1 injection, and you've got all the data and knowledge there to demonstrate that your method is still fit for its use. It still meets the requirements of the ATP. But you as a QC lab are going to save time and resources. And it's going to reduce the chance of obtaining an OOS measurement. That's valuable. And something that often happens with our example, let's look at what if the QC laboratory is asked to improve. Manufacturing isn't happy with the method. Somebody wants the QC lab to improve this method. Where should they look? Step 10, that sample extraction. Remember, it had 10 milligrams per gram contribution to the uncertainty. So you could either improve the sample extraction or replicate at the sample extraction step. Remember, S divided by the square root of N. And so here, if you look at it, if you took 2 test portions all the way through, and that means 2 injections at each there, of each sample prep, well, each uncertainty is decreased by 40%, square root of 2, so you can see down below. And there, the uncertainty combined becomes 7.6 milligrams per gram. It's about 8 milligrams per gram compared to the 11. Now, this is a lot of extra work. Is it worth it? Is that decrease of 3 milligrams per gram uncertainty around the reportable result valuable? Maybe you could do this and at the same time change to 1 injection for each sample prep. That would get rid of some work, and we know that would have no impact on the 8 milligram per gram. So you decide. But as a QC lab now, you've got data and probability to decide what you want to do. So looking at the analytical process with its units, sample prep, measurement, replicate strategy, you can come up with control strategies for each of these, as we did, that sample prep. The measurements, are you going to do 1 or 2 injections for your replicate strategy? And you can develop your control strategy. So finally, that quality risk management process, all these requirements, this is a standard risk management chart. For an analytical procedure, you can use the evaluation and use of measurement uncertainty to meet the requirements of this. Identification of uncertainty components, determining what that impact is, and looking what you're going to do, and how are you going to reduce that with your analytical control strategy. So, in conclusion, you can be efficient and effective with your analytical procedure using the life cycle approach. You're not going to waste time and resources. And you can confidently defend the performance of that to any regulatory agency. And the test results will support manufacturing. They will be fit for use. Step 3, continued procedure performance verification is for the quality control laboratory. That concludes this part of the presentation, and we're now ready and open for live questions.
And thank you, Jane, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located at the far left of your screen, and we'll answer as many of your questions as we have time for. So Jane, let's take a look at the questions already coming in from our live audience. What are all the common problems that QC faces?
Thank you. QC can face a lot of problems, and common ones, really, when you come down to it and investigate, you find out that they weren't told all the knowledge about the test method, the analytical procedure. Maybe Pharm Tech or the R&D group knew there was an issue, dealt with it, but didn't communicate that. And so that is the source of a lot of problems, and that's why knowledge management is important in the life cycle approach. Another example is the control strategies not adequate. Often the uncertainty is not evaluated correctly or at all, and it turns out that a part of the method can have a huge impact on the uncertainty, and it's not evaluated properly. And it's the QC lab that finds that out and has to deal with it. And again, the life cycle approach with a control strategy gives the QC lab the knowledge and capability and tools to deal with those. And so on a high level, those are some of the problems that a QC lab faces.
Thank you so much, Jane. And one of our audience members wants to know, can you give us the exact differences between a QA and a QC?
Okay, quality Q assurance. All right. QA is quality assurance and QC is quality control. And the definition of the difference between them often depends on the industry you're in. In the pharmaceutical industry, they are very strictly defined, and the FDA and the CFR, for example, uses quality control to cover the whole quality field, including what's called quality assurance. Quality assurance in other industries is the department that has the responsibility and oversight for quality for whatever that company or industry is doing. And quality control is just the laboratory that does the testing. And so those are some of the differences between quality assurance and quality control.
Thank you, Jane. And again, I want to thank our audience for their questions. Jane, this next question is about certification. Should an analyst be able to prove that they are certified to verify instruments such as balances and pipettes?
That's an interesting question. And coming from the ISO 17025 accreditation world, with the new standard that came out in 2017, they called the analysts competent. And you have to demonstrate their competency, and you have to authorize them to do an activity. And so if an analyst is going to be verifying a balance or verifying a pipette, the QC lab manager has to make sure that that person knows what they're doing and can do it. And they have to provide adequate training, and they have to, at some point, authorize the person to do that. There's many ways of doing that are very practical, but they have to be authorized. Yes.
Thank you. And Jane, do you think a biochemist has the capacity to become a QC?
Yes. Often the people who work in quality control labs come from very many different fields. I, for example, was a classically trained inorganic chemist doing fire assay in the mining industry, and was able to transfer to a quality control lab in pharmaceutical with no problem. Again, referring to the previous statement, question and answer about the competency of the analyst, with a QC lab, they should have the proper system to make sure their analysts are competent, and along with that would be reviewing the skills, education, training of the person. Often in biochemistry, they have some lab work and some knowledge, and so that would be applicable to becoming a QC person.
Thank you. And is the QC also the analysis the capsulation of tablets. I think they're asking, can they also analyze the capsulation of tablets?
I'm not quite sure what you mean by the capsulation of tablets, but QC labs do analyze all kinds of products and finished products and in-process products. So if during the capsulation there were some samples taken for in-process, yes, they could do that. The QC lab, no matter what they're going to be analyzing, requires a qualified test method by the Lifecycle approach. And so with that, if it's, say, capsulation, then the pharmaceutical technology department and all that should have decided what needs to be tested and why through the quality by design, and given the QC lab the appropriate test method for those capsulations of tablets.
Thank you, Jane. And we have time for a few more questions, and I want to remind our audience that any questions not answered today will be answered via email. What is the status of the USP draft General Chapter 1220 on the life cycle?
Oh, that's good. I'm glad you asked that because I was going to say something and I didn't. There is a new draft article coming out, a stimuli article, and it will be published in the USP Pharmacopoeia Forum. So watch for it, either the next one or the one after that that's coming out. And so you can read that. It's the USP's latest thinking on the life cycle approach, and you can comment on it. So you can become part of the writing of that. You can comment on it, and the USP does, and they have to consider every single comment that is given. And I just want to say with that, the USP has created a new general chapter expert committee called Measurement and Data Quality. It's just being created right now. And it will be responsible for taking that Chapter 1220 to make it official. So keep watching the USP website and that for information on that life cycle approach.
Thank you, Jane. Our next audience member wants to know, how do freshers get their experience? Because commonly nowadays only experienced members get their job.
Well, that goes back again to the QC lab has to make sure that their people are confident. And so if they're going to hire somebody with less experience, they can do that, no problem. They just have to make sure that the QC lab provides the needed training and education that person needs. And as well, it is recognized, there's a number of ways of doing that, and it is recognized, especially in 17025, the ISO standard, that if you're going to have somebody with less experience in that working on an analytical procedure, they can do so, but they need to do it under the supervision of a qualified, competent person. And so the QC labs can bring in less experienced people as long as they have the system to make sure that they are competent before they test actual samples.
Thank you. And regarding FDA, where does the FDA reference the life cycle?
Yes. Yeah, I mentioned a bit of that. If you read through the guidances on the FDA's website, and a couple of years ago they came out with one for the analytical procedures, they do mention the life cycle. For example, in the life cycle for manufacturing, they state that the analytical procedure must be suitable for the stage of life cycle of the manufacturing process. And also, a lot of FDA guidances mention that the risks have to be controlled. That, again, is an indirect reference to the life cycle, which is based on the risk analysis. And FDA guidances also mention things like they have to be appropriate, significant, impact, all these words. That means that you, as the lab company, have to know the impact, in which we just saw in this presentation. When you evaluate the uncertainty, you know the impact of each step. And so the FDA is referencing life cycle more directly now. If you look in older guidances, they actually were saying it too at that time, using risk and significance and words like that.
Jane, what's the materials and labor hours cost per test?
Oh, that all depends on the test, right? You can have a very simple loss and ignition where you just need a furnace at an oven at 110 and a crucible, through to a highly difficult biological ELISA with antibodies. So that the cost and all that can vary a tremendous amount upon that test. But that's an interesting question, because a lot of my presentation, if you went back and you had the cost of each of those, and often when I was in the lab, I had to know the cost of each of those steps, and the cost of doing an injection in the HPLC, I knew that, and then I could use that amount. So if I were going to decrease from 2 chromatograms to 1, because it had no impact on the uncertainty in this example that we had, I could calculate that. And so knowing the impact and the cost of the method, the uncertainty can help very much with that. So there's a whole different variety of tests and costs, but the uncertainty with the significance can help you understand and quantify that.
Absolutely. And how difficult is it to estimate the measurement uncertainty?
Well, I don't find it difficult at all, but I have a lot of experience in doing that. So, what I found out is that it really isn't that difficult, and that most laboratories have most of the information they need. The benefit of going through and evaluating the uncertainty, it tells them which information they don't have, and then they can go get that. And sometimes that means you just go back and look at old data, and sometimes it means that you have to do an experiment or two. The vast majority of times that I go into labs, I find they do have the data, and I can estimate the uncertainty in a couple of hours. And so with experience and with a proper life cycle approach, it evaluates the uncertainty as part of what it does. It's not that hard and doesn't take that long. There is a learning curve to it, but eventually you can get much faster.
Thank you, Jane. You shared a lot about how the materials vary from one another. How does the drug synthesize every time maintain quality?
Okay. In the example that I meant more of a lot of reagent in the laboratory. Sometimes the reagents are difficult to obtain, or they could have an impact and you only have one lot. That's what I was talking about. What's the impact of changing a lot of reagent to a test method? But here you were asking a little bit about using a lot in a manufacturing. So that is different, of course, but that also happens there. So they're manufacturing and they have a lot of some reagent, a catalyst, for example, and eventually they're going to run out of it and have to get a new lot. And they will have to manage that change as well. And so, the quality by design is being applied in manufacturing, and it's being applied in the laboratory. And so in both places, you have to know what's happening, understand what's happening, control what's happening. And so for that, understand what's happening. Oh, we've only got one lot, and we've never changed lots. We better be careful. And control what's happening. Okay, we're going to test this new lot really carefully. All right. And we're going to watch the first product we make with this new lot, and test it a little more. So that's an example of how you can look at the different lots in your manufacturing by quality by design.
Thank you, Jane. We have time for one more question. Our audience member says, I am a biotechnology master's and have experience in biochemistry and chromatography. Is there any chance of getting a job as a QC person with my background?
It sounds like, yeah, with that kind of background, you would have some of the knowledge and experience. Chromatography is a commonly used technique in quality control labs. And so you'd have the understanding, the scientific understanding. I don't know if you have any experience with quality control, but there's a lot of good courses out there on quality control, quality assurance. You could read the USP papers to start to understand what that life cycle is about. And along with that additional information, you would have most of the qualification you need. I would just like to refer again, the quality control labs are required to have programs, right, to select the appropriate person and provide the training and education that that person needs in their lab, and to assure that person is competent before they actually start testing real samples.
Jane, thank you again for your presentation today. Would you like to provide any final comments to our audience before we close?
Oh, thank you for that opportunity. Just one comment is, this life cycle approach, I'm so excited about it. Having worked many years in quality control labs, and I look at the life cycle approach and they have some aspect and say, oh, only we'd had that 20 years ago. Or, yes, I've had that problem, and this part of the life cycle approach would have prevented that problem. And so I encourage everybody to read about it and watch the USP website and provide comment and feedback as we all go forward in implementing this new approach.
Thank you again, Jane, for your time today and for your important research. We'd also like to thank LabRoots and our sponsor, Laboratories, for underwriting today's educational webcast. Before we go, I want to thank the audience for joining us today and for their interesting questions and questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. I'd like to once again thank Jane Weitzel for her time today and for her important research, and thank LabRoots and our sponsor, Sartorius, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, be safe, bye-bye.